factors that are affecting the chance of success. So the sperm, they have to traverse this different distance, and they're going to do that by swimming. So they need to be a good quality sperm, and there need to be enough of them that eventually some of them would be able to make it that long distance up to the ovulation. Uterine muscle contractions, this can happen within the female that help move the sperm to different places of the female reproductive tract. But then over and above these sort of physical things, there are also some other cues and events that happen that seem to facilitate fertilization. So one of these that's fairly recently discovered is that there exists a temperature gradient within the oviduct, where it's only a very slight difference of 1 or 2 degrees, but the sperm can sense this difference in temperature and they'll swim up the oviducts to where the egg is located. And I think it's slightly warmer up there in the oviducts. In addition, it's long been thought that there may be some chemotactic events as well. So either the egg or the granulosa cells may be secreting some chemical cues that could then, basically diffuse in a gradient fashion. If the sperm sense these, they would know what direction they should swim. They try to swim towards the source of higher chemotactic cue. So both of those might be used by species, including humans, to try to attract the sperm to where the egg is within this relatively large outside world. Then another thing that was noticed when people started to manipulate fertilization events in Petri dishes, so we could collect some eggs and we could collect some sperm and put them together, and it was not very successful. And it became appreciated that the sperm inside of the female reproductive tract actually undergoes some kinds of changes that are a bit mysterious. They're called capacitation, and what it reflects is that the sperm somehow become a little bit more able to engage in fertilization successfully. And what this probably involves is that there are different molecules or cues inside of the female reproductive tract that are acting on that sperm in some fashion and enhancing its ability to fertilize. So capacitation is a general word that refers to all those processes. So we have then about 100 to 300 million sperm ejaculated. About 200 actually reach the site where fertilization occurs. So your author, Scott Gilbert, he has a joke that if the sperm would just ask directions, that maybe more of them would get to the final point. But, actually it's kind of a small number of sperm when you think about, all that huge number, so maybe that's the point. Hyperactivation. This is another change that sperm go through in the female reproductive tract, where they become more active, that is, they're swimming more vigorously. And so that's obviously an important part of being able to fertilize, is to swim vigorously and get to that final destination. Thermotaxis, that was the attraction based on chemical cues. Chemotaxis is the attraction based on chemical cues. So temperature, if I said that wrong, and chemicals. Okay. All right, we had a diagram that looked like this for sea urchin. So now we can revisit the story with what we know about mammals. We know that the egg structure is a little bit different. So there's no jelly layer here. Instead, what we have is just your ordinary oocyte surrounded by a membrane, so the egg cell membrane. Outside of that is that clear layer, the zona pellucida. It's made of glycoproteins. And then outside of that is the cumulus layer. So this is really a layer of cells, granulosa cells. And I don't know why they just draw it as a yellow bar here, because that's not really very representational of cells, but cells is what it is. So sperm, when they're coming along, they're going to have to push through this outer cumulus layer, push through the zona pellucida, and eventually you still want the sperm cell membrane and the egg cell membrane to encounter each other and fuse. So the sperm, they have this acrosome, and they will undergo an acrosome reaction. And this happens when the acrosome encounters the zona pellucida. So in sea urchin, it happened when it encountered the jelly layer on the outside. And so it's just a little bit different structure here, where the acrosome is going to, again, exocytose its contents. And in principle, the same kind of thing happens where these proteases now digest their way through that zona pellucida. And then we're able to have the sperm cell membrane and the egg cell membrane fuse together in successful fertilization.